So I would like to thank Jimmy and Jairo for inviting me to participate in this symposium. And it's great to be talking about perspectives and advances in arachnids in the neotropics. And I have the great pleasure of talking about my preferred group of organisms, Amblypygids. So I'll be talking about systematics and biogeography of neotropical Amblypygids. And so I will start with a small introduction about diversity and distribution of the group. So Amblypygi has 5 families. We can say 5 families: Phrynichidae, Charinidae, Phrynidae, Charontidae, and Paracharontidae in the whole world. And they are Amblypygi is one of the small order of arachnids, so they are little diverse. There are 224 species. And of these families, only 3 of them are found in the neotropics, which is Charinidae, Phrynichidae, and Phrynidae, which total 130 species. And these are the genera that are found here in this whole area. So Carinus, Heterophrynus, Paraphrynus, Phrynus, and Trichodamon. And there has been a great advancement in the knowledge, on the taxonomic knowledge of the group in the last few years. So for example, in the last 10 years, there has been 46 new species described in the region for all these different genera, which is an increase in 130% compared to the previous 10 years, which for the group is amazing, it's a lot. And here are some examples of groups of species that were described recently, like Carinus gutu from the Amazon, Carinus tingomaria from the Brazilian Amazon, this is from Peru, and Heterophrynus origami from the Amazon also. And there has been a lot of advances in taxonomy, but not as much in the knowledge of the evolution of the groups, behavior, biogeography. But I will show here some progresses that we have made, like I mean, I'm not including in all the papers and projects that I'm showing here, but like I have done some works and collaborators and other colleagues have done other splendid works. And so I will be showing here some advances in systematics and biogeography, and also show in the end perspectives of prospective works that could be done. So I organized in a systematics and biogeography section and then a phylogeography section, and I will show each of the families, and I will show like progresses in papers and things that have been done. So we start with Charinidae, which is the largest family within Amblypygi, and it has more than 90 species, which is a large family for like in the Amblypygi world, the Amblypygi scale. And it's very widely distributed. It's all over the world, all over the place. And the genus Carinus is the one present here in the neotropics, and it has the widest distribution. And recently there was a paper that I studied and that I worked in my PhD. And what we have done, we have sequenced some sequences, the usual suspects, some loci, and we made a phylogeny, molecular and morphological phylogeny to try to understand relationships of all the groups. And what I'm showing here in this zoom in part of the tree is are the clades that include species that occur in the neotropics. So what we found is these clades, Oceania and South America clade and Eastern South American clade. The Oceania South American clade includes this one clade here that we called cubensis, that is basically species from the Caribbean and Central America. And this group is sister to this other clade that includes species from here, Carinus insularis and Carinus cupcake, that are sister to species that occur in Australia or in Oceania. So we propose some hypotheses of how this relationship could happen, but there are other taxa, other groups that have this kind of patterns also. And in this other clade here that we call the Eastern South America clade, we have this clade, which includes Carinus silami and the new species that we are describing from this area here, that are sister to the species that occur in the eastern part of Brazil. So these two are related here. So this area here is the one with the highest diversity in the neotropics of Carinus. It has more than 30 species, and it's the Atlantic forest. So most of you probably know that the Atlantic forest is a highly diverse area. It's a hotspot of biodiversity, and this is also reflected with Amblypygids in that area. And right now I'm doing a postdoc trying to understand why that area is, why that region is so diverse within Amblypygians, and what were the processes that led to this high diversity in the area. But there is still a lot of work to be done with this genus, Carinus, in all these different places in South America. So we dated the phylogeny, we calibrated with only 2 fossils because there are not many fossils of Amblypygids, differently from spiders that Daniele just showed us. But that's the best that we could do. And we got these dates here, and here are the clades that includes species that are in the neotropics, and it shows how ancient they are. And they are in South America and other parts since like 168, 148 million years ago, which is since Gondwana was present. So they were in this big landmass that since then split. And the wide distribution that we see nowadays of Carinus is probably reflected from the presence of these populations that were distributed in this big continental plate back in the days. And it's the same for what we see in the neotropics, because they can, as I showed here in this other slide, they can go from the tropics here up to here. So completely between the tropics, you know. So for Amblypygi, at least, it's a large distribution. So this is what has been done for Charinidae recently, and now I'll show you a Phrynichidae, the other family that we see in the neotropics. So this is Trichodamon princeps. It's a beautiful animal. It has these really large pedipalps. And Trichodamon is a genus that we see here, we find here in northeastern Brazil. And it's the only genus in this family, in the Phrynichidae, that occurs in the neotropics. All other genera are either African or here in the Arabian Peninsula or in Asia. And there has been no studies to date when this split happened or to place phylogenetically where this genera fits within the Phrynichidae tree. So in 2018, I published results from from my master's that I did in Rio, and first we did some exploration of a revision of the genus Trichodamon. So it has a wide distribution. It goes, so this is northeastern Brazil. This is like the dry area of the Caatinga, which is a dry area in the country. And it has a huge distribution. By the time when I started working, there was 2 species known from this area. And we were trying like to delimit the species morphologically. This was all morphology work. So we were trying to find characters that could separate the species, but we found a huge variation in morphology and color. For example, the species here on northern distribution, they are brighter than the species in the southern distribution, and there's this decline of colors between these 2 areas. So we could, using morphology, we couldn't differentiate the species, so we lumped them together. Now there's only 1 species, we synonymized the 2. And then we made a morphological phylogeny ranocladistic analysis to place the South American genus within the family tree. And we found it as sister to Phrynicus and Euphrynicus, which are genera that are in the eastern part of Africa. So yet we couldn't date and understand a little bit better the biogeography of the group because we didn't have molecules for them by that time. But this is something that needs to be pursued for sure. This is an area of research that needs to be done. And it's surely very interesting. We'll find, anyone who does this will find interesting results. So moving on to another family, Phrynidae. So here we're seeing a mom of a natural Phrynis carrying its babies. Very beautiful picture from a very beautiful specimen from the Amazon. And Phrynidae is very widespread also in Central America, part of South America, and a little bit of North America. And who knows a little bit of Amblypygids know how crazy it is to identify them because there's so much variation in spine number of the pedipalps. There's so much variation in genitalia morphology. And a few years ago, Daniel TDV, he took the challenge of doing a PhD studying this group, and he revised the taxonomy and did the phylogeny. It's not published yet, but he authorized me to show his data here, and he'll be publishing this very soon. And he tried to understand like homology of spines on the pedipalps and the different parts of the pedipalps. And he created a large morphological matrix and also sequenced a bunch of specimens. And in Phrynidae, we only know, nowadays, we know these three genera, Phrynos, Paraphrynos, and Acanthophrynus. But according to the data that he produced in his PhD, there's actually 5 new genera that he creates. And for example, here is Acanthophrynus coronatus. It's a genus with 1 species only in Mexico. And according to this analysis, actually include way more species. And the same thing with Paraphrynos and these other genera that were created that actually took species that were in Phrynos or Paraphrynos. So basically Phrynos and Paraphrynos is a trash can where people don't place anywhere, just throw them there. And hopefully this will be published soon, and will help guide us to understand better the evolution of this group. And in the same work of Daniel, he also showed these maps with an updated distribution of the genera, because they have this wide distribution, and the species sometimes they look so much alike. So it's good to have an updated view of where the species are to help us guide, also have a better understand on the group as a whole. So now I'll move on from the phylogeny and biogeography, which help us understand like big scale relationship between groups. And I'll move down to population level phylogeography, which also help us to understand, delimit species and understand how many species are there, because it gives us a better way of assessing cryptic species, for example, and subtle differences between groups when you go down to the population level. So I will start with Charinidae again, and this is a work I've been doing here at the Smithsonian as part of my postdoc. And as I talked before, I've been focusing here in the Atlantic forest studying Carinus. So this is Carinus montanus. And this is part of some results of the project that I've been running, part of the results. And this was focused on the mountain region of Espírito Santo. So this is a zoom in the Atlantic forest. And there is several populations of Carinus montanus. You can see here, montanus. And there is a species that is sympatric here in this area. And we wanted to understand how is gene flow among populations, if there is gene flow among populations, or if actually there is cryptic species over there. We wanted to understand a little bit better how gene flow happens within Amblypygids and specifically within Charinids, because we knew nothing before. So for this, what we used was UCEs, ultra conserved elements, so genomic scale data that we sequenced. And this is a relationship that we found. I'm not going into details of methods, but we can, if anyone wants to know more, we can discuss afterwards. But this is the tree that we found, the relationships of the populations, like these populations are more closely related to each other than this. And this one is sister for all others, but there's actually no more clear morphological difference between any of these populations. And there is great genetic separation, differentiation of these populations from the others, which is very likely a cryptic species. And then we run some genetic analysis, some population genetic analysis to understand if there was shared haplotypes between groups. And actually we found that there is that the populations and the specimens in the populations, they were very distinct. There was not actually a lot of shared haplotypes between groups. So the redder, the more similar they are, the yellow, the more different they are, and they are mostly similar to what they are from the area where they were already collected. So moving on for another group and another region, we are seeing now the family Phrynidae, the genus Phrynos, and this was a work done by Esposito et al., published in 2015. And here they studied 4 species in Puerto Rico and Dominican Republic. So Phrynos longipes, Alejandroi, Eucharis, and so 3 species. And they found like great genetic structure which actually follow geography. So we see here, these are the limestones found in the island. And so the populations found in this area, they were highly differentiated from populations found in this area and following this area. That's what we see here, North Sea limestone, central metamorphic rock. So what they claim is that there is like islands within islands, because these species, they occur inside caves. So although they're big and they can move quite well, they don't go around actually. So they don't disperse and they don't encounter other populations, and there is no gene flow between them. And they suggest that the amount of cryptic species is way higher than what we expect for these species that occur in caves within the islands in the Caribbean. And that is the pattern that we are finding also for other groups in South America, as the Carinos that I just showed you. Another recently published work was Sater et al. 2020, published very recently, that they used Paraphrynos in Mexico. And they included several species that occur in the area and in the Caribbean. And they also found one cryptic species that occurs right here in the middle of the distribution of the other species. They call it Paraphrynos pseudomexicanus, that is sister to Paraphrynos mexicanus. They used a bunch of genetic markers, the usual suspects, and then after they found the genetic differences, they went back to the specimens and actually found a few morphological differences, like size of the frontal process that is smaller in the new species compared to the other species. Several other differences regarding number of spines on the pedipalp, trichobotria on leg 4. And they also went on to extra molecular details and analyzed karyotype, and then they suggested some evolution of the differences of karyotype between Phrynos and Paraphrynos, which is something that I don't dominate as much, and I invite you guys, anyone interested, to read the paper if you want to know more about evolution of karyotypes. But it's something interesting too, and new for Amblypygids that is not done much within this kind of methodology. And another group that had recently been explored in a population level was Heterophrynos, also published this year, Reveillon et al. And they wanted to understand in Heterophrynos these 3 species, Alcis, Patesi, and Longicornis. They wanted to see, they collected in different areas, and they wanted to see if there was gene flow, if there was hybridization, or if there was cryptic species, isolated cryptic species in different areas. And they actually found great genetic structure, as we see here, Heterophrynos longicornis from this area and Heterophrynos longicornis from other areas of branching. This is based only on one gene, but still it's a guide, or is something that it shows that it's something that should be explored further, both morphologically within the group to explore more, to see if there are differences, and also molecularly, including more genes to understand better how these species move and hybridize or reproduce and get in contact with each other. And yeah, so talking about CO1, which is what they used here, there is also barcodes. So another work that was published in 2018 was within Heterophrynos in Peru. So they sequenced the barcode of several specimens in this area. And then they took sequences from both that were available online, and they wanted to see genetic differences. They wanted to see if they could use the DNA only to tell apart which species that was, which can be risky if the taxonomy of the group is not very well established or very well known. But it's something to be done as a starter, or it's something that it's good to have also more genetic data available online, so other people in other regions can compare. So it's not ideal if you only do this, but it's something useful. In the more we do, the more information we will have, and then we'll have more confidence on the data that we get regarding barcode.
Más o menos 20, ya pasamos 20 minutos, te aviso.
Okay, I'm in the last slide now. So just to talk a little bit about open questions and perspectives, as Daniele said, with spiders, there's a lot to be done still with taxonomy and distribution of the group. Here's a map showing like large gaps of areas. There are no dots of Amblypygids known. whole countries like Paraguay, large countries like Bolivia, and still here Colombia, Venezuela, and also Brazil. So there's still a lot to be explored, so we understand better where these species are, and also understand better their morphology and their evolution. And other open questions that needs to be explored is why some areas are so much more diverse than others, like Charinidae here, Phrynidae here, something that could be joined with a historical and ecological perspective, so we have a better whole understanding of the evolution of the group. Regarding Phrynidae, the group that Daniel Chirivi did his thesis, we still don't have a dating for that phylogeny, so we don't know when those branchings happen. And this species, Phrynidae exu, it's so all Phrynidae are here in the neotropics, and there is this one species that is in Indonesia, and we have no idea how it got there, when it got there. So this is also some really interesting line of research that needs to be explored. Again, cryptic diversity, Amblypygids, most of them look a lot like the other, they look alike. And there's a lot to be explored there, because when species that are widely distributed that we think is one thing might actually be more species. And also the use of new methods like UC, micro-CT, evo-devo, that will be shown actually some evo-devo with Amblypygids in this congress, and barcode, which is not a new method, but it's a new method within Amblypygids. And these are things that will help us have a better and more thorough understanding of the evolution and diversity of Amblypygids. And with that, I would like to thank everyone for watching, and thanks my collaborators, my funding agencies, and all the places that facilitated research with Amblypygids. And thanks again for inviting me for this symposium.